Welcome back. In this module, we're going to start to introduce multivoxel or multivariate pattern analysis. And I'll first talk about some of the neuroscientific principles and underpinnings that'll help us understand why MVPA works and why it's a good idea. So traditional brain mapping approaches treat the brain as the outcome and the task or behavioral variables as predictors at each voxel. So they test at each voxel whether there's some non-zero effect, but they can't actually tell how big that effect is. It's just a hypothesis test. And the brain is always the outcome. Well, so this means that every voxel is a separate, independent outcome in a mass univariate analysis. There's no models or no accounting for interactions between or even joint additive effects of various different brain regions in representing or predicting outcomes. So this is a major limitation, and often what we like to do is we'd like to use multiple voxels to explain an outcome because we know that brain regions work together. So that's what multivoxel pattern analysis does. It assesses whether a pattern across brain voxels predicts a behavior or an outcome. So the goal now is to identify patterns that are sensitive and specific to particular outcomes, and then we can use those patterns to test the relationships among them and understand something about how the brain represents those different outcomes. So MVPA is multivariate in brain space, unlike the standard mapping analysis, and it's often, but not always, univariate in the task or behavior or outcome space, whatever you're trying to predict. So there are some exceptions, but most analyses are univariate in outcome space. We're trying to predict one outcome. And this kind of analysis, I would argue, is better suited for mapping brain structure to function. Why? Because we can develop models of interactions among brain regions that characterize a predicted category of mental events. It turns out that I don't know any single voxel in the brain that's really sensitive and specific to any particular category of mental events. So that approach is really limited, and this one is better suited to this mapping procedure. So there are two main types of multivoxel pattern analysis, and the first type I'll call local multivariate analysis. So this means identification of patterns in one or more local region, like a region of interest, and then looking at that region and asking, does V1 predict the outcome? Does V2 predict the outcome? And this also includes so-called searchlight analyses that take a local region and a moving window, and they just run this predictive analysis everywhere in the brain, like that. The second type is what I'll call an integrated model. So in this case, we're going to use voxels across the whole brain, any available information that I think is neuroscientifically valid, and sometimes even multimodal data, like combining functional data with structural data, fMRI, EEG, PET, or other kinds of data into a single predictive model. So this is really suited for asking, how well can I predict this outcome using the available brain information? And then I can refine the model, simplify the model, and ultimately work with that as a basis for a representation of the underlying process. So why would we want to use MVPA, apart from the reasons I've already told you? One is to identify these patterns that are optimized to be predictive of external outcomes. That could be experience, performance, clinical status, etc. And then, even if I don't know exactly what fMRI activity means in terms of neural activation or inhibition or where exactly the signal is coming from always, if I can predict an external outcome, essentially my brain results have their own internal logic and validity, and it becomes its own viable level of analysis for predicting performance. A second reason is to better characterize the mappings between mind and brain for the reasons I said before. Because really, I don't know of any single voxels that really encode categories of mental events and can really be used as indicators for them. So this MVPA is especially useful for capturing structure at finer spatial scales than typical voxel-wise analysis. We'll look at that. And it's also really useful for capturing brain representations that are distributed across voxels or regions or even networks that span multiple brain regions. So let's look at the issue of spatial scale. And as I said in the previous course, there's information at multiple spatial scales, going from large-scale networks, which have some differences across different types of psychological categories, to functional maps and regions at a finer spatial scale, to functional columns at a still finer spatial scale, and then finally cell assemblies at virtually the single neuron resolution. So there's information about what kind of task and outcome processes are happening at all of these scales. And we looked at some examples of somatosensory maps and of head orientation maps, all within a millimeter or two of cortex, and among others. Other examples are ocular dominance columns and orientation columns, which again, their spatial structure is quite fine-grained, often smaller than the voxels themselves that we are assessing. So if we think about what standard bold fMRI is sensitive to mapping, it's really only to the large-scale networks and to some degree some of the larger functional maps. But with MVPA, we can push this boundary and we can extend into these finer-grained maps. And we can do this because we're capitalizing on patterns of information. So just another note on why do topographical maps form in the first place. So there are some properties of the brain, the physical structure of the brain, that afford the development of these topographically clustered regions of neurons. And one of the properties, the neurons have short-range excitatory and inhibitory connections. And what I've drawn here is two neurons with an excitatory axon collateral that connects them. So this is a pretty common structure you see in the brain. And that's going to produce similar representations and receptive fields for similar neurons in similar locations in space. And the second property is experience-dependent plasticity. Neurons that fire together wire together, so to speak. If you put those two things together, and this is an example of a typical sort of center-surround organizations where we have activation, excitation in a local region around a neuron, and then inhibition farther out. If you put those properties together with learning, the fire together to wire together property, then you can end up developing networks that develop a spontaneous topographical organization. And one example of this is the Cajonin network. And what you see this cactus here is this example illustration of this model that was trained, and it starts with a random set of response properties across a different field of so-called neurons, and through experience, because each neuron excites its neighbors, it tends to learn similar things as its neighbors. And then similar areas of space then start to represent similar areas of, in this case, three-dimensional geographical space. So in the MVPA literature, why can it work? Why can it actually detect these finer spatial scale organizational structures? And there's this idea called hyperacuity, which is this idea. So voxels, a typical voxel at 3T is about 3 by 3 by 3 millimeters, pretty big. And each of those voxels might sample, on average, say 5.5 million neurons, many of which do different things. So the distribution of the neural types, if you will, in a voxel might be mixed, but across voxels, the precise mix varies. So this simulation here illustrates this. This is a homogeneous Poisson process. It's really it represents a random distribution of neurons of two event types, so two with code for two different properties. So if I could measure every neuron, it would be really easy to distinguish when the organism is engaging in event type 1 versus 2, just see which neurons are active. But of course, we sample these when we sample them in voxel space on this very coarse grid. But now we're getting an average. And the idea here is that some voxels, even if there's a random chance distribution of the voxels, some of those voxels will contain more neurons that respond more to event type 1 than 2, and other voxels the opposite. And so across the pattern of voxels, I can actually detect some differences. And whether we can do this in any case is an empirical challenge, an empirical question. But this is an example that often real neural structures are organized like this. So this is from Daniel Salzman's lab and Joe Payton, and they're looking at electrophysiological recordings in the amygdala in monkeys. And what they see is intermixed neurons that respond to positive events and negative events. And the distribution doesn't seem to be clearly separable. So it looks kind of a lot like the homogeneous Poisson process that I've diagrammed here, perhaps. So here's an interesting property. So even if the neurons are intermixed randomly, then MVPA might still be able to identify patterns that are distinctly and reliably associated with each. So here's our two intermixed types of neurons. And the point here is that random doesn't mean identically distributed, doesn't mean completely homogeneous. And so what's happening, these neurons are active, and what the bold signal is really picking up on is local field potential. So now we've smoothed these neurons into a local field potential representation here for event type 1 and event type 2. So now you see the topography is still a little bit different. There's a lot of information there. And then when we sample that in our discrete voxels at low resolution, we can still see that there are different patterns of voxels across those two event types, even though the two event types were randomly intermixed. So in fact, the voxel patterns are largely uncorrelated. Here in this example, R equals 0.15, so not very highly correlated. And it's possible to classify the type of event, even though the intermixing occurs at a much finer spatial scale than the voxels themselves.